Yeah, thank you very much for the introduction. I'm very happy to be here today with you. Thank you, Paul, for the invitation, and thank you, Danielle, for the perfect organization. I will present to you today some of my research. I started in the mid-90s and did a PhD in the field of art history. I handed in Berlin 2003 on art and biotechnology, which was in those days in the art fields quite familiar, but not in the field of art history. And I will show you today some examples, especially for those who are not that familiar with BioArt, and then come to a more recent projects I curated or co-curated, and then to the question of the conference on the question of the materials or the materiality of BioArt, and what does that mean for institutions like art institutions or disciplines like art history. Is this a kind of new art form, which is actually not that new anymore, but is this an art form that will be part of the art market, that will be part of the curriculum of the art history institutions sooner or later? And I think the question is yes, but then if BioArt or artists working with living materials will be part of the more I would say traditional art fields like art market or museums, art museums for contemporary art, then probably the education system has to adapt, or and of course curators and institutions have to go in that field as well, and of course some do already in an extensive way. So in the 20th century, the 20th century experienced a radical change in the production and as well in the perception of art, and saw during the 20th century the rise of many new art forms, of course accompanied by a considerable change of framing and also explaining art. With the rise of what we understand today as the classical avant-garde, embedded in an age of mass industrial production, numerous materials became employed by the arts. Even waste materials became appropriate and integrated as material and a source of aspiration. I show you here one example, Kurt Schwitters, which is quite famous, and of course it brings a headache to all curators and to owners of these artworks. And it's quite interesting because all these most of the elements Schwitters is using he picked up from the garbage, and of course these materials were never meant to be part of the museum archive. So, and it's quite interesting, there's a German insurance company, AXA, which is one of the biggest insurance companies for art museums, and of course they have big problems because these artworks were never meant to be part of the art institutions like museums. And so it is quite difficult, and this is just the beginning. This is the beginning of the 20th century, and of course it will be even worse when it comes to BioArt, and when you see have cells and tissue culture and things like that. So these problems are already out there, and so even waste materials became appropriate and integrated as material and a source of inspiration for the arts. And to quote my friend, the Mexican philosopher Maria Antonia González Villario, in that sense, aesthetics, art history, and art theory and critics had to reformulate their concepts and theoretical frameworks to meet the changes posed by contemporary art. So the artists are the one, or the movements in the arts force the institutions to adapt and to change. So with the emergence of BioArt in the late 1980s, biotechnology and genetic engineering became also part of the art world. By this means, living entities such as bacteria, cells, tissue culture, DNA, and transgenic organisms moved from scientific laboratories into galleries and museums. Of course, there's a long tradition in the 20th century art concerning the use of organic material like meat or blood or sperm or even living animals. But with BioArt, the perspective on organic material changed significantly because BioArt looked from a very different perspective, and from the very beginning looked at scientific advances which are increasingly challenging our concept of life and our perspectives on a techno-scientific futures in which, above all, life probably, or life itself, will be human-made. So I show you now some examples. One of the first ones by David Kramers, this was back in the 90s, in 1992. He already worked in those days at Caltech in the States and was able to work in a lab with colleagues from the biotechnology department, and he was one of the first artists using genetically modified, I think it was bacteria, E. coli bacteria, and and then there were other artists following. This was an American example. This is a German, Edgar Lissel, who is based in Vienna for a couple of years. He also started to work quite early. This is a later artwork I'm showing you now. He started quite early to work with cyanobacteria, but he's a photographer. He's, like traditional educated in photography. But because these bacteria have kind of photo effects, I would call it. He started to work with bacteria in the lab, and also went into, this is Edgar when he was, about 10 years ago. And he went into the lab and worked with bacteria, but on quite traditional artistry topics, or I would say artistic topics, like the self-portrait, about the question of representations and things like that. So he is still, he used in those days cutting-edge technologies, but had, like in his mind, a more traditional approach towards these new, in those days, new technologies. And what is important, why I show you these two pictures here, it was necessary for him to collaborate with a scientist. Nowadays, a lot of artists have their own little labs at home or at art school, but in those days, he had to find someone in the science field to collaborate, to get access to the laboratory to do what he wanted to do. And another example in Australia, so what I show you here is Peter Clancy. She also used bacteria and other organic materials in the laboratory. This is her from the back. And but she tried to produce like representational images, like something that we would understand as like a human being, a human figure. But then she showed it in the white cube in the gallery in a different way. So it was produced by bacteria, but then it was shown like a project, like really something you can like put on the wall, and so it was able to get access or get inside a traditional art gallery. And then there were also artists beginning like Christine Borland from Scotland. And this was quite interesting because she already started in the 1990s working with HeLa cells. I don't know if you're familiar with HeLa. HeLa is a very, very important model organism in biotechnology that was, I wouldn't say invented, but found in 1951 in the States when an African-American woman, Henrietta Lacks, therefore it is called HeLa, had cancer, and so this was one of the first immortal cell lines that were discovered at the right time, at the right moment. But in 2010, a lot of books, or two important books, were published on African American women in the medical system of the United States, and all of a sudden HeLa and the whole situation about producing HeLa cells and identity and so on and so forth became a really vital moment. And Christine Borland, long before, came up with these topics. But the thing is, why I show you this example is that she tried to show part of the laboratory equipment, which was actually not the state of the art, because the state of the art would have been far too much expensive. Nowadays, microscopes in research institutions cost about 250,000 euros, and this was an ordinary microscope you could get, or she could get for free, and but still, she wanted to show that there is the thing itself, that she wanted to show something beyond representation, whatever that means, but this was the intention, and so sooner or later, more and more laboratory equipment moved from the lab into the white cube, into the galleries, and later on also in the museums. And of course then artists, they are well known, Oren Katz and Ayonut Zur, they try to show more complex cell structures, working on tissue culture, which has nothing to do with biotech, but is more or less a way of bringing cells into a form the scientists want them to be. So nowadays, tissue culture is a very, I wouldn't say basic, but it's in Berlin, for example, we have a few institutions producing tissue cells for people who have heart problems or people who burned their skin. So this is a technology out there. But when they started in the 90s in the States with an art and science resident program, it was really new that artists went into the lab, started to show things like that in the art context. So. And this is another a picture which is shown quite often, because then you can see how it looks like in the beginning, and then how the cells move and develop. And they used another project also called Disembodied Cuisine, and it's quite interesting. They started, I think, also in the 90s with this program. And showed it for the first time in 2000. And nowadays we have, for the first time, especially in the Netherlands, institutions working with in vitro meat, almost to an extent that it is, part of the food market quite soon. So these artists started working with or collaborating with scientists quite early on, long before it was on debate. And this is quite the last artwork I want to show from Ayonut Zur and Oren Katz, which was shown in 2008 at MoMA, which means that because MoMA is a very, I wouldn't say strong, but a very influential, powerful institution in the field of contemporary art, and whatever happens at MoMA, a lot of people are convinced that this is the right thing to do. Well, but if you ever have the chance to show an artwork at MoMA, you can be sure that it will be part of the debate. And BioArt were there in 2008. Of course, the interesting thing is that after a few days, it didn't work out because a gallery is not a laboratory. So all these bacteria got contaminated, but still. The photos there, it looks beautiful, and there is an extremely vital discourse, but this would be a very good example, and I'm sure a lot of people wrote on that since the exhibition 2008, because it was the first time that for at least 3 days a complex setting like this worked in such a prestigious institution, and a lot of people got familiar with that. So, of course, there's another materials like DNA. This is also like an icon in BioArt. This is Mark Quinn. a well-known British artist, and this artwork is quite, a lot of people are familiar with it. And here he showed the DNA of Sir John Edward Sulston, who was in charge for the Human Genome Project in Britain. And it was commissioned by the National Portrait Gallery and then exhibited in 2001. So this topic showing not sperms but DNA, because in the 60s very often sperms were shown as the thing itself, like a portrait, like an identity beyond representation. And now we have, in 2001, we have BioArt showing DNA, but the thing is, actually you do not see a lot. So. And here another example, Mark Quinn is using also his own blood. So he used I would say he's not a typical bioartist, but he uses a lot of own bodily material, which is, I think, part of the art world since the 50s or even earlier. So, and then there are other interesting artists like Paul Van Nuys, who also uses DNA in a debate, in a post-colonial discourse, showing his DNA and the one of his father, his mother, and his sister, and combining that, because he's from a mixed family, combining that with debates on early questions about race, where his family, part of his family background was grown up in the 1920s, where debates were vital, and research was done on primitive people from a Western perspective. So he's bringing that up, and he shows it in the gallery. And actually, because Paul is quite familiar with all these technologies, and the technology is not that cutting edge, things always worked out very fine. So he also showed in the gallery, in the museum, actually parts of things going on before in the laboratory. So this is like art moving into the art world. And I want to show you a few other artists, one German, one Austrian. They also went into the laboratory, but they were not interested in showing the thing itself or manipulating life and producing new organisms no one has seen before, but they wanted to know about the epistemological questions put on the agenda when scientists started to alter nature in a way that was not done before by evolution. And this is Rainer Maria Matusik, a Berlin-based artist, and he went long before American artist or Brazilian artist Eduardo Katz into the lab, working with people on transgenic plants. But he decided not to turn into a scientist or an artist showing science in the galleries, but he wanted to know what kind of consequences these developments in the sciences would have for art and for art history as well, and for his own perception of art. And this is really interesting because it is so early. It was in 1993. And the thing is, like, he made films, he made interviews, he produced new sculptures. He got very much acquainted with biotech procedures and protocols, and also went with his, this is him in the middle, he went with his students, so the art students also got acquainted with that, but he never was, keen showing these things in the gallery. He still stayed with his more traditional language of art, that is sculpture. But for him, it was important to be familiar with these things, and also for his students. And when he went out there, he still made sculptures and produce kind of representations of what he was thinking the next future step could be, but he didn't want it to show, really modified organisms. This is, I think it is Keynote. What do you say in English about Keynote? It's a kind when children play with how you say this? It's a very cheap material he's using, children use when they play with these things. It's like little, it's kind of wax, so it's nothing transgenic, but it looks like something. At the first place you're not, or in the first glance you are not sure what you see, and it could be something that could be alive, that could be transgenic, but of course it isn't. For him it is more like like a power play between him and the observer. And of course he also went into the galleries, into institutions, and wanted to ask what could BioArt be like in the future, and what does it mean if we have post-evolutionary organisms, if we really produce organisms in the lab that are not part of the evolutionary ladder. And so for him, this is like a metaphor. Things would, look like a mess also in the arts and in the gallery as well. So here you can see some of the post-evolutionary organism who create some chaos in the art world. And we made an exhibition together in 2010 where we showed at the Museum for the History of Medical Science in Berlin, which was actually showing parts of the collection of Rudolf Virchow. Most of it was destroyed in the Second World War, but due to the bombing of Berlin, but some of the collection is still there. And we thought about the problem, if everything is possible, if you can take some cells and manipulate them and grow them in what shape and form you ever want to, how does how could organisms or sculptures of organisms look like in the future? And the interesting thing is we also talked to a lot of scientists, and when you go to the doctor or you go to a hospital and someone takes some blood or some cells or whatever, sperms or egg cells or whatever you donate or have to give for research or for whatever illness you have, the material belongs to the institution and not to you anymore. So this is quite interesting because we thought what happens when these, say in 10 or 15, 20 years it will be possible that some of your cells, if you donate some cells, especially if you have still a young body, then it's much better because the cells move more vital. But who really cares for these entities and what kind of ontological status have these entities sooner or later. So it is not just an artistic interference into a museum, because these things we see a lot in Denmark, in Sweden, in Germany. We have a lot of exhibitions in these institutions for the history of medical science, but it's also a legal problem, and these things are quite differently handled in different countries. In Israel, it is quite different to the States or in Germany because of course, in Germany things are really complicated because of the history of Mengele and his companions. So in Germany, you hardly can do anything with these things, but it's very different in Israel and different in the States. So, and we also showed some of bigger, like the future organism, how the future could look like. So people liked that a lot, but we're also a little bit afraid. And we also showed some tissue engineering, and of course a video how all these things were made. But I want to show you another artist, Herwig Türk, who was based for many, many years in Lisbon, but is actually now based at the art school in Vienna. And for him, it was like a little bit like for Rainer Maria Matusik. He went into the lab, worked a lot with scientists, and never ever presented the thing itself in a gallery. For him, it was important to work with people from the history of science department, to go into the laboratory studies department, and to understand what new questions will arise when these things will be part of the art world in the future. Actually, he never did that, but he wanted to understand the process, what was going on. And the first thing, what a lot of artists do when they go into the lab, they make a photo of the refrigerator, because this is where it happens, or this is where the magic happens. But he also made a lot of photographs and called them escapes instead of landscape. And because here he, of course, was influenced by debates by Donna Haraway and other thinkers of reinventing nature in the laboratory, and what would be, like the future of art and also of science if we produce nature in the laboratory ourselves. So this was an important debate in I would say, in 2000, 2010. Was, like Bruno Latour, Donna Haraway, thinkers like this were read by a lot of artists, and then they somehow mirrored it back in their work in going into the lab. And he also makes fantastic, huge photographs like this one. This is Labscapes 5. This is a lab for flies, for Drosophila melanogaster. I hope I pronounce it right. But this is also one of the most important model organism. And for him, it was just more or less the aesthetic of how a laboratory looks like. And he never showed the things like this in the gallery. Actually, Mark Dion did that once, I think, in the Netherlands in an exhibition. He transformed the whole laboratory into a gallery, but Herwig only showed a photograph. And he also does a lot of videos and interviews, and it's about producing sculptures with laboratory equipment, asking the scientists to think about their own, about their own everyday handling, and the idea that there's theory, there's science, but there's also producing, there's the making of science as well. And of course, it's all about hierarchies in the laboratory, who, like the professors write the concepts, and then a lot of helping hands who are not mentioned in the end in the papers produce, like the work at the work bench. And then, of course, we have bioartists, very famous now, Eduardo Katz, that tried to work with transgenic organisms, also showing transgenic organisms. And I only show a few of his artworks because for me it is quite interesting to show that when I first met him, it was in 1999 in Brazil, in Sao Paulo. I think it was on a conference where he showed this GFP K9, where he wanted to produce a glooming dog. And in the same year or a year later, he showed his famous installation Genesis at Ars Electronica, and then only a year later came up with Bunny, which is, like I think one of the most debated artworks or bio artworks in art history. Actually, I would say so. It's only 2 years from a concept to something that is or looks like real, or the same here, only 1 year later, the 8th day, where he really showed transgenic organisms, like here transgenic mice. Of course, he did not produce these animals himself, but he was the first artist showing them in a gallery. And of course, this made a lot, because of the materiality, or showing living organisms is not that easy in art institutions like contemporary art spaces. So this I think only 2 or 3 years from the concept to showing the animal or a transgenic animal, and it worked out. Actually, the context he showed it were always it wasn't MoMA, it was institutions like art institutions in academic contexts like universities, exhibition spaces. So. And this is one of his latest projects. I do actually not really understand what he was, up to with that, but he combined his DNA with that of a plant. And this is maybe something about agency, or who is the plant, who is the human figure behind that, or who? Maybe it's because about authorship. I'm not sure, but this is one of his latest projects. And well, the question what rises here is what is art? Because people always ask, where's the art in BioArt? But of course, a lot of artists going into the lab ask what is nature and what is technology nowadays? And then the question, of course, what is BioArt? And some parts of the debates was about the context is producing the artwork. You know, like if you move parts of the lab equipment by an artist into a museum or into a gallery, then it is art, because the context is the instrument or the model that produces meaning. Or, and this is what Maria Antonia González Velario asks a lot and is much more competent than I am on that question, is BioArt a question about ontology? And then we have the debate, we have the object, and then we have the manipulated artifact. But of course, a transgenic animal is not an object. It's also not an artifact. Maybe it's in biofact. And so a lot of philosophers in the field of history of philosophy and philosophy and technology come up with this new term. What do we, or how do we handle, how do we perceive transgenic animals, for example, that were produced or made due to human technology, but are grown due to natural ways of being. So, and I want to show you a few other artists in the second part of my talk, because what I showed you now were a few examples of bioartists who are very much into this field of we look at biotechnology because biotechnology is able, since the 70s, to alter nature. Alter nature is something that is a big thing, and of course, art history or the artist, since the antiquity is seen as someone who wants to be better than nature, who wants to make the artwork alive and things like that. And all of a sudden, you can, via bioart and biotechnology, you can produce living art forms. But things are not that easy. So, and there were other artists out there who were more, I would say, more reflective about that mass production and mass pollution is already altering nature in our landscape and in our rivers and so on. And there is an American artist, Brandon Ballanger, with a wonderful French name. His family is from France, so but he's based in New York, married to a French girl actually. So and he went out into nature and looks at animals, amphibians, who are altered due to pollution in the rivers or in nature, and so the DNA gets somehow how you say in a way works in a way that it's not that for example it has 3, 4, 5 legs, that things are not the way they should be. And he is collecting these animals, showing them in different styles. These, for example, I show you here, are pictures of that size, but he also shows them the real animal after he is changing the skulls and the bones in about that size. So he has different media options to show these things in public. But he's also a scientist, and he was very much involved in the Deepwater Horizon oil spill in Mexico, and but tried to work or to communicate his artworks in a more traditional language, showing the thing itself, but like you see in a museum for the history of or natural history museums. And he went out there, and we met this year, and we went together to the Gulf of Mexico. And he was so much into that, problematic field that some of his research that he did was even used in a trial in the States because BP is not paying as much as the government expects to recover or to rearrange the damage they caused. So this is not only BioArt, it's not about altering nature, it's about more like, I would say, activism or artists who are also very qualified in the field of biology and science and laboratory studies go out to see that nature is altered and that there are many organisms extinct already. And and he just as an example, because I like it so much as an art historian, because Charles Darwin was quite important for art theory and art philosophy in the end of the 19th century, and Brandon is also one of these artists very qualified in the field of the history of science. He also went into a museum and worked on the some breeding experiments of Charles Darwin and found some of the birds or the pigeons he used, which were forgotten for about 150 years or 140 years, and he made artworks out of that. So it's not just about going into the laboratory and playing around with producing new organisms the world has not seen before and showing that there's a very mighty and powerful technology. It's more about being more sensitive about altering nature through mass production and environmental pollution. And what is interesting here, that he refers to Charles Darwin, because with Darwin we have, I wouldn't say a problem in art history, but with Darwin it became clear that species are never stable, that they change all the time. And this is a problematic thing, because if we say, well, we should not alter nature, we should not pollute the rivers and our landscape, then others could say, well, but things in evolution, things change all the way anyway. So there is no stable species, or the species are never stable. There is a constant change. But the argument is, or the problematic argument here is, what do we think nowadays when we use the word nature, or we should not alter nature in a political discourse? And so this is quite interesting that Brandon brings up all these topics, and starting in 1859 with the origin of species, and then of course with the whole debates on Lamarckism and things like that in the late 19th century. And of course, I show you another younger artist. She's from Turkey, Pina Yoldas, but she's living for many years now in the States, and she worked also in the field of bioart, but she has the same direction or the same idea like Brandon Ballanger, showing that even our oceans are altered and changed to an extent that all the bioart from the 90s going into the lab showing we can do this and this with bacteria is quite harmless compared to what's going on with plastic pollution in the oceans, because through this plastic pollution in the oceans, the whole ocean and the bacteria living and the organisms living in the oceans completely changed to a degree someone in the laboratory never could do, because the debate is always like, should we allow these altered organisms being part of our community? Should they be allowed to be released? The debate about Eduardo Katz's Bunny 2000 was all about, should these animals who are not produced in a natural way, or were not born in a natural way, although they were born but they were not produced in a natural way, should they be part, or should they be part of our society, yes or no? And this was only in 2000. Nowadays it becomes clear that the planet is already messed up to a degree that there's just not a question, should we allow them being out there? They are just out there. And hopefully they will not develop into organisms that eat plastic, for example, because then things get worse. But they are produced, or they they were brought into being because of plastic pollution. And this is just one example I show you from an exhibition we did in Berlin at the Ernst Schering workspace. And this is a kind of, she calls it plastic soup. This is 2 or 3 liters of water from the ocean. And to her, I think it is quite interesting, because for me, this is like a next step of BioArt. She is producing sculptures, so she's back to a traditional way of producing artworks with plastic and other materials. And she's showing or imagining a future organism that will be able to adapt to such a toxic environment we have produced in the oceans. So, and what is interesting, that this was the most successful exhibition. I will show you some more. So this is another organism, a future organism that will adapt to plastic, and plastic is colorful, because plastic is like what we have here. There's always, colors like blue or red or whatever. It's Pantone, and the Pantone colors, they will in the future show how nature will look like. And she made a couple of those ones. And it's interesting that her exhibition was one of the most interesting and most successful exhibition we had so far at Schering, because she was not showing the thing itself, not some little bacteria in a petri dish, but futuristic organism, which is kind of cynical. But on the other hand, a lot of people could get much closer to that topic, which is a very serious topic. And she collaborated a lot with scientists, and everything she did was based on knowledge delivered through interviews with scientists. And the interesting thing is, maybe because it's about her Turkish background, I'm not sure, but the thing is, if everything is possible, if we can produce all new kinds of organisms and change the DNA in the way we wanted to do it, why not enhancing? It's all about human enhancement. Why not enhancing, producing vaginas that are much more sensitive than anything that nature produced through natural selection or sexual selection, or which is kind of fun. But on the other hand, she's showing that, and people just love it. It's very, very funny, and she's producing wonderful artworks that were not produced in a laboratory, who had never seen any biotech instruments, but because of the visual language and because of the concepts behind what she has in mind, and because she is very, very qualified, also in the science field. This brings this topic to an enormous broad audience, and she has, I think we met 2 years ago, and then she just started with exhibitions, and now she has exhibitions all around the globe. And I think this is interesting, because I wouldn't say it's very different from BioArt, but it has a different approach, a quite different approach. How much time do I have now? How much? 10 minutes, okay. Because when we talk about materiality and art history and what is BioArt all about, there is, of course, what I showed you now was more traditional BioArt works that still produce something, that show something in a gallery or in an art space like MoMA. But of course, there is a quite interesting development that bioartists produce a lot of, workshops, invite other artists or lay people or curators, getting familiar with biotechnology, and very often there's no result like a product or something you can hang on the wall. It's more but it's a lot of fun. It's a lot of I wouldn't say gossip, but you learn a lot, and it's quite interesting. And I only show you that it's 2004, it's 10 years ago, but bringing the laboratory from the scientific field into the art world, into the art school, and then also into the kitchen is a quite interesting movement. And I would say 10, 15 years ago, a lot of people were not familiar with BioArt, but nowadays a lot of people even do do-it-yourself biology courses. So this topic is more familiar now than here in 2004. And of course there are do-it-yourself biology communities very often in art spaces. Like Gene Space, I visited a couple of times in New York, or other spaces. So this is a nice combination between artists and people who do community labs or set up community labs, and then also make their money by giving art courses at, for example, here people from Gene Space at SVA, School of Visual Art, which is super expensive, but for some money you also can do BioArt hands-on lab courses in summer. And because our debate was about how is art history or the art institutions reacting on that, this is not quite sure, but there are many art institutions that go into that field, making available access for art students. And here, for example, in the States, of course, for art students who pay a lot of money. And then there's, of course, another, I wouldn't call it art, but another movement, do-it-yourself bio, where a lot of community labs where quite often bioartists are involved as well. Like here in, I'll show you one example by the Waag Society in Amsterdam. And they have very interesting workshops going on. I think they just started last week, the Biohacking Academy. And of course some bioartists are on board as well, but what they also just opened at the museum, at the zoo in Amsterdam, is a new little museum, which is actually not that little, on microorganisms, that also have studios for artists going there, talking to the scientists, also producing an artistic outcome. So this is what's going on, and just one example, which I think is quite interesting, to bring up another topic here by a Swedish architect, Magnus Larsson, because especially in art history, it was very important always that art has no purpose. L'art pour l'art. This was something that for the whole 20th century was something that made art. And everything I showed you here in the field of bioart is a little bit like playing with new technologies, or not that new technologies like biotech. But nowadays we see a lot of bioartists going also into design and architecture, and they are very much fed up with having no goal. They want to do something that is meaningful, not a meaningful artwork, but a meaningful, like here, a project building new adequate architectural structures, producing concrete with bacteria, for example. So, and I think this is an interesting, I wouldn't call it term. I think there are a lot of artists out there still making their art just in their own art world or their own worldview, but there are more and more bioartists going into other fields like biodesign, design, architecture, who want with their knowledge, and very often these bioartists are now trained also in these fields and are very interdisciplinary. They want to do something that makes more sense for people like here in Africa. Which is a project to produce concrete out of sand, because if you produce concrete, you need sand from the seaside. So there's a lot of sand grabbing around the world, and very often, like at the coastline of Africa, in the morning people wake up and then their beach is gone because someone were grabbing it. So yeah, and of course we have other big institutions like here Vincent Callebaut, who's trying to bring this up into another scale. And I would like to end, how do institutions react on that? And I only show you one institution with the so-called Pheno, an art center in Germany, in Wolfsburg. Probably nobody knows here, but a lot of money was involved. And what they also have is, of course, a bio lab, not for bio artists, but for lay people coming in, getting more acquainted with biotechnology. Of course, I was a little bit astonished. I went there quite early, I think between 10 or 11 or something, and everything was empty. And I made some photographs and I said, oh, cool, nice, I can make good pictures. But I waited for an hour or two and still nobody there. And it's quite different to the photograph I showed you when the art, like Ion Azur and Arne Katz made their workshop, and we see this very crowded gang in a laboratory. So people get more familiar with living materials through institutions like that. And when they go to a museum, they will be more, and see BioArt, or BioArt, they will be more familiar with that. But still, it's a way to go. Thank you.
Thanks. Okay, thanks a lot, Ingeborg. Now we have time for some questions. Okay, there's a mic over there. I remember you can ask in English or in Spanish, and then we will translate her, whatever you want. Okay, first, Fernando.
Hi. Can you hear me?
Yeah.
Yeah, okay. So, hi, thanks for the talk. Just a question of clarification. I don't know if you can expand a little bit more on the distinction that you made between context and ontology. I think that you put it that way. You said that the way in which people have understood BioArt is by saying that it's actually the context, the institution that gives meaning to these artworks, and then you opposed to that ontology.
Yes, this
but
is
I wasn't quite sure what you meant by that. So can you clarify what you mean by ontology and what is the opposition to context?
Yes. Yeah, unfortunately, Maria Antonia Bellario is not here. She would be the much more appropriate, giving a more appropriate answer. But the thing is, like, we talked a lot when I was with her last semester in Mexico City about the question, what is art and what is BioArt, and is BioArt the appropriate art form for the 21st century? And she combines this question with the question of ontology, what is art? And should it be connected to what Hegel said with zeitgeist? Should it be like a metaphor or something so we understand our own time a lot better? So for her, the question about BioArt is an ontological question, but for the most people writing on BioArt, it is a question of context, that it is not that important. The ontological question is not that important, but more important is the way in which meaning is produced in institutions like art institutions or museums, art history debates, curricula in art history and things like that. So the shift of context does not ask about the question if a living organism that is shown by an artist is already an artwork, or a living organism that was manipulated, that is, or it could even be not manipulated. This is what, like Duchamp did, just bringing something like an art trouvé, objet trouvé, and I'm not sure if this is an appropriate access to BioArt, but as far as I see, there are these two, maybe more than these, but these are two vital debates on how we perceive or how BioArt is accepted as art form. Because in the beginning, I would say in the 90s, people said, oh, this is not art. But this is the same, like in the 60s with media art, they said, oh, this is not art. But it is quite interesting that the question, what is art in the 21st century for us, is somehow debated, very vital in the field of BioArt, because a lot of people just hate that art form. and think this is not art, this is technology, or this is just playing around, or this is something that it has no tradition or there's no bridge to the traditional art forms. But this is not sufficient. But I would this answer is not sufficient to your question. But I would refer more to the philosopher Maria Antonia González Velario, because she extensively wrote on the question of ontology and bioart.
Okay, more questions over there? There's one there.
Thanks, Ingeborg. Can you hear me? Yeah, sound? Yeah. Thanks for that talk. I think my question is a little bit related to social ontology as well. I think you touched a little bit on it earlier, how some of these artists in the 90s were influenced by Donna Haraway and Bruno Latour. I was going to ask you to elaborate on how and if these artists, and many of the artists you mentioned today, or by our artists, do this art, or this becomes an art form to discuss and challenge how technologies change or erase or their distinctions between art and life, nature and culture, human, non-human, and so forth.
Could you repeat that?
Yeah, I think you have to put your mic more in front.
Can you hear me?
I'm sure it was super interesting, but
Yeah,
the question. But could you?
No, okay, I do this quickly again then. No, I was going to ask you. I mean, you mentioned earlier that some of these 90s artists.
Yes, Havik Turk and others. These were just two examples.
Yeah.
We were reading Latour and Haraway and Lily Kay and
Yes.
these laboratory studies and others.
Yes, I was going to ask you if you can elaborate on how these, or if these BioArt artists kind of aims to discuss how technologies change forms of life, forms of human beings, synergies between nature and object, and so forth.
Do you think they changed? Or what?
How nature changed.
Yes.
If that, a theme which they discussed, the technology's role within that change, or if it changes or not.
She's referring to Donna Haraway. Well, I'm sorry, but we cannot hear a lot what you're asking. But you were talking about the concept nature cultures by Donna Haraway and the production of nature. You're talking about that, how this relates with the bioart, and how this concept could relate with bioart. I'm right?
Oh yeah,
Okay.
yeah. There's no. I think I grabbed it now. But of course there are quite a few artists who did that, and it's interesting that these artists were loved by institutions like in Berlin, for example, the Max Planck Institute for the History of Science, because these debates or these artworks. I showed only two examples. There are many other examples. They were a wonderful way for scientists who are trained in the history of science to refer to contemporary art, because although I only can say it for the German context, the history of science is a wonderful field of research. It is not very well funded. There's an exception with the Max Planck Institute of the History of Science in Berlin. But referring to art, a lot of institutions like museums, small museums that were not very trendy in the 80s and the 90s at all. Who went in the 80s or 70s to medical history museums? If you go to, and now it changed completely, and all these museums have art programs now, like the Medical Museum in Copenhagen, and they are very happy that they have these artists at hand that fit perfectly in their debates, in their discourses, and bring new people or younger people into their museums to give them a new, I wouldn't call it, produce a kind of pop lifestyle for medical museums, which were, I wouldn't say boring, but they were very silent places I would say, up until the 80s. Not everywhere, but it's interesting that there was a kind of artists were referring to these scientists, referring to that literature, and then these people were inviting artists, producing artworks and concepts together, and then writing again about these artworks. So, I don't know if this was an answer to your question, but I think for some institutions it was the possibility all of a sudden to be more trendy and to also involve more people, because most bioartists or artists who were invited have a lot of knowledge also in communicating to a broader audience, who have a lot of knowledge about design, and so I think this is a very interesting connection between artists referring to this kind of literature, to that debate that was so much loved by these scientists, because by these, also people from the humanities, because all of a sudden they could refer to these topics, but in a more fashionably, more trendy way, and so.
Okay. Well, thanks. That's really interesting. I think both questions connects also with this ontological, let's say, ontological distinctions that fund categories and taxonomies, let's say, which is quite common in art history, where categories and taxonomies is sometimes the way. This ontological distinction also creates a different, those distinctions between nature, culture, and all these things. So how we in this way work these in-between distinctions, ontological distinctions between nature and culture, posing this nature-culture concept as a way, as something, culture, nature, not something that has to be explained, but nature, something that is being produced in bio labs or whatever. So this is maybe that's the last question. We are running late, so Danny, you have your last question.
Yeah, please. Well, I'm glad to hear your talk. I have a lot of questions, but I have choose two. First of all, as you know, I have been doing research on BioArt also during some years. And one of the things I noticed is that there were a lot of people talking and writing about BioArt, but there weren't art historians about this. You were the first one I knew working on BioArt. And most of them, they were artists, philosophers. And my first question is this. Why do you think this is happening, or this happens? And second, what can art history, maybe, I think it's not maybe ontological question, what is art history, but I wanted to say this. And on the second question, it's about what we have been talking also with Fernando, between ontology and context, because I think that BioArt has been already explained as almost like a ready-made, something that you take from science and you present in context of a gallery, and this makes this a work of art. And I think when we talk and when we oppose this context to ontology, we are talking, I think, about one topic of the Congress, about materiality in BioArt. I mean, I think the key question, at least for me, is what is the difference between living artworks and non-living artworks? I mean, between works that you have presented that use photography, not painting, but Dalí had paintings about DNA, and other works that use living tissues, cells, and I think there are some interesting ontological points that are arising with living artworks. I don't know if you agree or if you establish a difference between living artworks and non-living artworks in BioArt.
like Oren Katz and Ayan Azur talking about the semi-living, but of course this is a hoax. There's just it's life or death. That's it. But to your first question, why not that many art historians? I think there are some, but the problem was, or I think refers to the question how to make a career in academia, and the people I know I'm working with. When they turned to BioArt, there already were professors like Robert Swienenberg in Leiden University, who started in 2005 the Arts and Genomics Center. And the big question was, how do we, because we were talking a lot, because I just finished in those days my PhD, and my PhD supervisor was a brilliant scholar, but no help for my topic. And we thought, what would be an adequate way for young philosophers and art historians to get acquainted with BioArt? And what we decided was, with Jenny Bulbuli, who made an absolute wonderful PhD on BioArt, is sending her to the classes at the biology department, so she had to go into all these laboratory equipment courses for about a year, so she would know when she would interview artists slightly or better than we probably would know in those days what things are going on. So we really thought about how can a PhD look like for 3 or 4 years. And there are some art historians, but still, I guess it is too interdisciplinary. Most of my colleagues still go; they work on Richter, or they work on, I don't know, all kind of more traditional contemporary art, because they want to have jobs afterwards. And I think with PhD in BioArt, maybe because you were the first here in Spain, you will be fine. But so don't get me wrong. But yeah, and there were just not that many supervisors. But now I think things change, and now the students in Berlin, they love that topic. They love that topic, and they are also very interested in biodesign, in food and biotechnology and things like that. And I think at the moment things are changing, and it's also a matter of money, of course. And at Humboldt we just got a grant, 2 professors, actually not me, 25 million euros to go into biodesign and cultural history. So things change a lot. But the problem is that we just last week we had an enormous debate, because the way that our how you say in English, the protocol how to make a PhD at Humboldt-University is fixed somehow, and it is very difficult to bring people in and say, oh well, he has to go or she has to go for 1 year to a biolab, and then maybe she can come back. And it's the institutions are not as open yet to that. Therefore, I guess more artists like Susan Anker and many others, or Critical Art Ensemble, they were writing a lot more, and they did also art. PhD for artists in that way, which was more. But I think there are more and more people, and it's a topic that is growing. And
so okay, we're going to stop here because we have another session that will be also in connection with this one. So maybe we will have time later on to discuss about the whole thing. So thanks a lot, Ingeborg.